Ladies and gentlemen, slowly start finding your seats. We're going to be starting here in approximately 2 minutes.
All right, everyone, can you please find your seats and we can go ahead and get started with our next set of speakers. Okay, so to start off the second half of this session, we have Jason Neuswanger from WDFW, and he will be presenting on an integrated population model of Wenatchee River steelhead.
Hi, thank you. So today I'm going to be giving a first look at what's very much a work in progress still on a population model for steelhead in the Wenatchee. I imagine that these fish need no introduction here, but just in case you missed the first several talks, this is steelhead that spawn in the Wenatchee as part of the ESA-listed Upper Columbia steelhead population. In times of greater abundance, it used to support a bit of a sport fishery, but that's been closed down recently. We have estimates of returns from the Wenatchee and the age structure of this population going back into the mid-1980s. This time series is based on a wide variety of data types and analytical methods that have evolved and improved over time. Most recently, Kevin C. has been using patch occupancy modeling to produce the most accurate estimates to date. This population has been supported by stocking throughout the time period I'm analyzing, and we have estimates of both hatchery and natural origin spawners. You can see that there are major ups and downs in both time series. To look at what's driving them, I'm using an integrated population model. This is a type of model that combines two important components in a single hierarchical Bayesian framework that keeps track of uncertainty all the way through. The first component is the run reconstruction, which is basically an estimation of which spawners came from which generation of parents and vice versa. And the second is the population model itself, which reflects our hypotheses about the capacity and growth rate of the population, and whatever in the environment might affect those rates. The particular IPM I'm running lives in an R package called Salmon IPM, which is being developed by Eric Buell and is based on some work I believe he initially did with Mark Sherrill, who I think is here today. To analyze the population, we organize it by brood year, which refers to all of the offspring of parents that spawned in a given year. It's worth noting that the brood year is actually the year after the run year in this case, because these fish arrive in the fall of one year and then spawn the next spring. This chart shows which years relative to a brood year contains important milestones for a large population of, or a large portion of the progeny from that brood year. After the parents spawn in the spring of the brood year, the eggs incubate in the gravel, and then the fry emerge and begin growing that same year. They will spend up to 3 years growing in the Wenatchee and its tributaries, and most of them will smolt and enter the ocean at the second to third year after the brood year. Those that survive in the ocean will eventually return home to spawn or try from the third to the fifth year after the brood year. A life cycle that spans so many habitats over so many years comes into contact with a huge number of potential influences. And I'm using the model to look at a bunch of these. This is not an exhaustive or a final list, but it just gives you an idea of the many ways that things like temperature, stream flow, ocean conditions, and the hydro system can be summarized to investigate hypotheses about their effects on the population. For this talk, I ended up investigating 28 different metrics that weren't too closely correlated with one another. I computed all of them using weighted averages that reflect which portions of the life cycle would be exposed to which effects in which years. The most direct goal of the model is basically to predict the number of recruits from each brood year. That is the number of offspring from that year's spawners that eventually come back as spawners themselves. However, this is easier to interpret when looking at it per capita, i.e. the number of recruits per spawner. This plot is the number of natural-born recruits from each brood year divided by the number of spawners that produce them. We would ideally like to see an average of at least 1 recruit per spawner, which is called the replacement line. That's the point at which each spawner is likely to replace itself with at least 1 offspring in the next generation. Unfortunately, we're below that threshold most of the time. To approach this mathematically, we can plot the number of spawners in each brood year against the number of recruits. Here, each dot represents spawners and recruits for a single brood year, with error bars around it reflecting the uncertainty in those estimates. The horizontal replacement line from the previous plot shows up as a diagonal line on this plot, and years with good recruitment should be above and to the left of the line. I've color-coded these by PHOS, which is an acronym for the percent of hatchery origin spawners. The red dots are years when most spawners were born in a hatchery, whereas the blue dots had mostly natural origin spawners. We have lots of red dot years with relatively high numbers of hatchery origin spawners, but very few recruits. Blue dot and purple years with more natural origin spawners tend to fall closer to the replacement line. Because of this apparent difference, I'm alpha testing a version of salmon IPM that allows for different population parameters between hatchery and wild fish. The blue curves here represent the predicted number of recruits for a given number of spawners under scenarios with 80% PHOS in red or 20% PHOS in blue. Each curve is defined by 2 important parameters. The productivity parameter alpha controls the initial slope on the left-hand side of the curve, and the capacity parameter Rmax controls where the curve levels off. From these curves, we can see the model is suggesting that the capacity of the system is quite a bit higher when we have mostly natural origin fish. But there is some uncertainty around these curves in a way that can't be reflected by looking at a single line like these. Next, I'll show you what that looks like for an example with PHOS of 0.5. Actually, that's going to be on the next slide after this one. We're actually representing the productivity and capacity parameters as probability distributions with a range of possible values weighted by how probable they are. Red shows the parameter distributions for hatchery origin spawners and blue for natural origin. On the left, you can see that there's almost no difference in the estimated productivity parameter between the two, but the capacity parameter on the right is substantially higher for wild fish. However, there is quite a bit of uncertainty around that estimate because we don't have very many years with high numbers of natural origin spawners. If we were to show the uncertainty in these parameters reflected in the individual stock recruit curves, it would look something like this, with this black line being the model for PHOS equals 0.5, and the heat map around it reflecting the probability density of where the most likely true position of that curve should fall. So just keep in mind as you go through looking at the single curves in this talk that there's actually this kind of baseline level of uncertainty around all of them in the background. So now we have a model of the relationship between spawners and recruits. But it's hard for, sorry, I just realized I got a little behind on the slide there relative to what I'm looking at. This is the slide that shows where the uncertainty is. So now we've got this stock recruit model, but it's pretty hard for a simple set of curves like this to convincingly fit such a scattered set of data. To understand what's driving this scatter, we're going to start by looking at the residuals from this model. The residuals, which are plotted here over time, are what's left over of log recruits per spawner after accounting for whatever variation was explained by the previous model. In years with residuals above 0, the fish produced more recruits per spawner than the model expected, and vice versa. There are some clear long-term patterns of ups and downs here, and it would sure be great if we could find some variable that very closely tracks the variation that the previous model couldn't explain. You know, like this yellow line, for example. We are in luck because this is actually a real time series called the North Pacific Gyre Oscillation, or NPGO. This is an index relating to ocean currents that strongly correlates with nutrients and chlorophyll in certain parts of the ocean off the west coast. So in a nutshell, it's a way to look at the productivity of the ocean when and where the steelhead smolts are arriving. Others have identified this index as potentially important to steelhead on the Skagit and on the Olympic Peninsula, and there's a very strong signal for it here in the Wenatchee data. An important caveat with here in any of these covariates is that there are often some other things that kind of track the time series in a similar way, maybe not quite as closely, but could potentially present themselves as alternative explanations for these effects. In this case, some of the indices of Nino effects and ichthyoplankton biodiversity kind of track the same thing and could potentially be a part of the actual explanation for what's going on, but the model just kind of goes with the best one here. It's satisfying to find an explanation of some of the variability that fits so well. So we're going to try it again, but first I want to talk a little bit about how I'm selecting variables to include. Because this is a preliminary analysis, I followed kind of an intuitive ad hoc process just as a placeholder for something more rigorous in the future. I added one covariate, starting with the NPGO, and then reran the model. Then I looked at correlations with residuals from that model and added the best one of those to the model and reran it, and I just repeated this until I ran out of interesting relationships. After building in the NPGO and then looking at the residuals from that new model, another strong effect turns up. This is the average time it takes water to move through the hydro system during smolt migration. In years with longer transit times, the fish did worse than the previous model would expect, and vice versa. However, this variable is also related to the average spill percent through the hydro system during smolt migration, so we might be seeing a signal of either or both of those effects. The bottom line is that we seem to see better recruitment in years when smolts have a quicker and easier trip down through the 7 dams and associated reservoirs that they have to transit to get to the ocean. So now we built that one into the model and look at the residuals again. And the next thing that shakes out is an index of the nearshore abundance of ichthyoplankton in the Pacific in the winter before steelhead smolts are going to arrive in the ocean. Running a little low on time, so not going into too many details about the rest of these, but we've got, after we incorporate that effect and look at the residuals again and look at the next thing, it comes out that there's a negative correlation of wild recruitment with the number of hatchery smolts during the years that the wild fish are smolting. We're definitely getting into correlations now where you can see a pattern, but it's a little bit fuzzy and they don't look as convincing, so you take the relationships kind of from here on as pretty speculative. This one in particular warrants attention because it's kind of similar to what's been reported for steelhead in the Skagit as well. Next, we have the median winter flow in the Wenatchee across the years when the juveniles are rearing, not the maximum flow. So this is not an effect of fish getting blown out by high water. If it's real, it's probably something to do with poor conditions for growth during periods of sustained high cold water. But it could also be something less direct, like a signal of ocean conditions that determine rainfall, that determine stream flow. The next effect is the timing of the physical spring transition, which is related to upwelling that brings nutrient-rich water to the surface in the nearshore parts of the Pacific around the time that the smolts are getting to the ocean. The later this transition happens, the worse the steelhead do relative to model predictions. Whoops. Lastly, Tom DeGroza brought to my attention some previous work he did on the Aniat River, which showed that juvenile steelhead were benefiting from eating summer Chinook eggs. This model is at least hinting at a population level signal of that effect in the Wenatchee, but again, kind of faintly. With all these covariates in the model, I went back up here and revisited what the model looks like without them, just as a point of comparison. For the next slide, I'm going to remove the modeled effects from the various covariates, or from these data points, and replace them with what the model sees as purely the dynamics linking the number of spawners to the number of recruits. Pay attention to how the curves change over that time, too. This is what we get. Much of that scatter in the data is gone, having been accounted for by subtracting the proposed environmental effects. We still see a higher capacity when more of the spawners are wild fish, but the difference is less than it was before. Now, the one last thing I can do with this is look at the distributions of the parameters associated with these covariates. These are the distributions for the environmental effects. Long story short, the colored areas represent 95% credible intervals, and the farther they are from the vertical white line, the stronger the effect and the more likely they are to be real. There are a lot of caveats associated with this, but the bottom line is that there are pretty compelling signals for ocean conditions and the hydro system affecting Wenatchee steelhead recruitment, and there are at least some interesting possibilities in the freshwater environment in the river as well. So what's happening next with this? We've got a lot of good data on smolt abundance that I'll be able to incorporate into a more complex version of the IPM. Going to be looking at more environmental covariates and figuring out a more rigorous way to incorporate them and choose between them. We're going to expand this model to other systems and integrate, like, for example, the Aniat and the METHOW at some point in the future within the same framework. And lastly, we should be able to eventually use this tool as a bit of a predictive tool to explore different environmental scenarios and management scenarios moving forward. So I just want to thank a bunch of people who've been involved with the modeling and the data collection, and I think I'm out of time. So.
All right, next up we have Ben Goodman, also from the Washington Department of Fish and Wildlife. His presentation on evaluation of the reproductive success and fitness covariates of hatchery, wild, and reconditioned Kelt steelhead in the Twisp River.
Thank you. So I'm going to provide some brief background and the latest results we have from our steelhead reproductive success study. So we have three main research questions, which I'll address in this talk. First, do hatchery steelhead have lower fitness than wild steelhead? This is important because we're releasing hatchery fish to help recover these wild populations, and if the hatchery fish have lower fitness, then we could be reducing the fitness of the wild population as well. Kelts are being released or reconditioned throughout the basin, and there's some uncertainty about their reproductive fitness, so we want to know, how does Kelt reconditioning influence fitness? And finally, do phenotypic and demographic variables explain any differences we see? So our study area is the Twisp River, and the main thing I want to point out here is that there's a weir at river kilometer 11 that constructed by Douglas PUD that allows us to capture adult steelhead. And we also have a smolt trap and pit array down near the mouth that allow us to capture and detect fish. So our first objective in the study is to estimate the relative reproductive success of hatchery steelhead. So we get hatchery and wild fish at our weir in the Twisp, and we release them upstream to spawn naturally, and then we estimate their reproductive success at 3 different life stages, par, smolt, and adult. And then we use genetics to determine who the parents and offspring combinations are. So due to concurrent hatchery reforms, the source of broodstock for the Twisp program changed during the course of this study, more than once. But so we started out having what we refer to as well stock, so hatchery by wild crosses of fish collected at wells, and switched to a local Twisp stock collection of wild by wild fish. And so I'll be showing some results from those. So these are our genotyping and parentage assignment results, and you can see that we've been keeping the genetics lab pretty busy. The main thing I wanted to point out here is that we're missing a lot of parents. So only 25% of the potential offspring we get have both parents assigned. 63% have at least one parent assigned, and most of these were mother only. So we're missing a lot more fathers than mothers. The source of these is likely from contributions of residents and hatchery residuals. There's also a fair amount of spawning below our weir, and we sample for offspring down there. And in addition, when the water gets high, the weir gets a little leaky. Anyway, so now I'm going to show you some of our relative reproductive success results. But first I want to make sure we're on the same page about what that is exactly. So the relative reproductive success of hatchery fish is just the average number of hatchery or of offspring per hatchery spawner divided by the average number of offspring per wild spawner. And I'll show some, they'll have confidence intervals that are from the profile likelihood method. So here we're looking at the relative reproductive success of hatchery females from Twisp and well stock. And at all the life stages we measured. And so what you'll notice is that all of the juvenile estimates, or the majority of them, are below that dashed line. Anything below that means they produced fewer offspring than wilds. So at all the juvenile stages, we were generally seeing a lower reproductive success. For the adult to adult portion, it's a little more variable and much more uncertain. And that's because the sample sizes are a lot smaller. And those are still, some of those estimates are still preliminary, those adult to adult ones, because we're still getting adult returns that have yet to be analyzed. Another thing I wanted to point out here is that you see that the 2014 year is kind of the one anomaly here in terms of the juveniles. So in that year, it was the last year of the well stock returns, so they were pretty much all 2 salts. And length is typically correlated with higher reproductive success, so that explains why we had that bump there. And here we're looking at the relative reproductive success of the hatchery males, and we see really similar patterns. The juvenile estimates are generally showing a lower reproductive success, and the adults are kind of bouncing around and have a lot of uncertainty. And here, the one that stands out is a little bit different is 2015 for the Twisp stock. And what's likely going on there is that was the year we had the lowest proportion of wild males in the study. And so there was just less competition for these hatchery males. And so here we're looking at pooled estimates, so all those years just pooled together. And I wanted to show this mainly because we don't really see a broodstock effect. So in other studies, in the Hood River and here in the Wenatchee, they saw that if a higher proportion of wild fish in the broodstock resulted in higher reproductive success. And so far, we haven't really seen that. All right, so our second objective here is to estimate the relative reproductive success of reconditioned kelts. So starting in 2014, the Twisp wild females that were kept for broodstock were incorporated into this Kelt reconditioning project run by the Yakima Nation. And essentially, they hold these fish in circular tanks in a facility at Winthrop National Fish Hatchery, treat them for fungal infections and parasites, feed them a rich diet of krill and pellets, and release them in the fall to spawn again. And the pictures we're looking at here that Matt provided show the same fish right after it was spawned, and then on the bottom is after it's been reconditioned. So for 2015 to 2017, we had 11 of these fish in our study. And the thing you'll notice here, in contrast to the hatchery fish, all of these estimates are at or above the one line, suggesting that these fish produced more, or as many or more offspring as the maiden spawning wild females. So that's pretty cool. And our last objective I'm going to talk about is evaluating differences in phenotypic and demographic variables that might explain what we've seen. So for phenotype, we measure the length of the fish, we take scale samples, and their arrival day, sex, and origin are all recorded. And in 2012, we started measuring somatic lipid content using a Distel fat meter. This was developed for the commercial sector to grade meat and fish fillets. And it allows us to take a measurement on a live fish. And then we did a generalized linear model analysis, pooling all the juveniles as the response variable, and added some annual covariates, only one of those per model, and modeled females and males separately, and ended up doing 64 models for each sex, basically all the possible combinations, and then used AIC for model selection. So our best models are shown here. The blue indicates a positive effect, red negative. So for females, length, lipid, and day all had positive, that's arrival day, all had positive effects on reproductive success, and being of hatchery origin had a negative effect. For males, lipids had a positive effect, and again, hatchery origin had a negative effect, as did sex ratio. So if there were, as the male to female ratio went up, their success went down. And what I wanted to point out here is that there's, quite a bit of uncertainty in the model selection, especially for males. Only 16% of model weight is in this top model. So here we're looking at the coefficients for model averaged coefficients, and the thing I wanted to show is this this RI, which is the relative variable importance. And so for somatic lipids, 98% of the model weight included somatic lipid content for males, and 94% for females. And fork length was also really high for females. So I'm just going to dig into a couple of these relationships. So here we're looking at the relationship between lipids and reproductive success on the left, basically showing that on average, fish with higher lipid levels produced more offspring. And then on the right, that's the differences among these groups, their average with confidence intervals for their somatic lipid contents. So the thing that pops out there is that those reconditioned kelts had really high lipid content, and that might explain some of their high reproductive success. In addition, both the Wells and Twisp stockfish had lower lipid content than wilds, and that probably explains some of their lower reproductive success. And here we're looking at fork length on the left, basically showing that on average, larger fish produce more offspring. This is just for females. And on the right, we're looking at differences in fork length among these groups. And so the one that kind of stands out here is those twisp stock fish. So they seem to be smaller on average than our other groups. And this may explain why the lack of a broodstock effect in our study so far. So the smaller size of these fish might have offset the benefits of the wild parents. And also the reconditioned kelts were larger than average too. So moving forward, we're continuing to work on evaluating the second generation relative reproductive success, so comparing wild fish with hatchery parents and wild fish with wild parents, which could provide evidence or lack thereof of a genetic effect. And then we've also incorporated Winthrop's age 2 smolts in our study since 2019. These are fish that are released at age 2 as smolts rather than age 1. And that's it. I'd just like to say a huge thank you to our collaborators, the funding agencies, and many, many coworkers over the last 10 plus years. Thank you.
All right, last speaker to close out this session will be Wesley Tibits from the Confederated tribe of Colville Reservation. And he will be speaking on migration estimates of hatchery summer steelhead may lead to early discrimination of outmigrants.
All right, well, thank you all for coming, or more importantly, thank you for staying. I know it's right before lunch, so probably all hungry. My name is Wesley Tibits. I work for the Colville tribes, and I'm their hatchery summer steelhead monitoring and evaluation project lead. I also want to acknowledge my co-author, Brooklyn Hudson. He did most of the heavy lifting on this project, so I want to give a shout out to him, as well as our funding agency, Grant PUD. The objective of our study was to better understand juvenile outmigration of hatchery summer steelhead in Omak Creek, which is a tributary to the Okanagan River. And we wanted to determine if there was any delayed migration and if it occurred, and to what extent. We also wanted to find out what environmental factors there might be that are driving delayed migration in summer steelhead. And what are some of the possible positive effects to delayed migration as far as a life history strategy? So what is a residual? I think most of you know what a residual may be, but I wanted to point out that most managers will estimate residuals within 3 months of release. So most fish are released in April at high flow, and so they're looking at June 1st to come up with a residual estimate. Often these have adverse effects on resident fish, and fisheries managers often predict those effects and monitor any potential impacts on natural origin steelhead. You can see some of the differences in size here for residuals. That bottom picture to the left, you can see small fish still having par marks, and then some of the larger fish might be really precocious and produce milt. Those are pretty good indicators of a residual steelhead. So what are some of the related hatchery studies? Locally, I tried to choose a few here. Age at release from Matt Cooper, Michael Humling, which looked at size and maturation. An immigration survival study, which from Buchanan and Skalski, which is kind of what we modeled our work from. Limited habitat and diurnal temperatures and day length. And then we also looked at effects from residual hatchery fish looking at more of the impacts from local fish, from Todd Pearson and others, and then ecological impacts and demographics of hatchery steelhead that are residual from Charlie Snow and friends. So where are we located? We're located in north central Washington. The Okanagan River is kind of the last place for steelhead to migrate to spawn in the lower 48. And we're located on Omak Creek. You can see the little red dot is Mission Falls. That's where we release fish. And then they go out to the Okanagan and then into the Columbia. So a little bit of history or background with Omak Creek. Omak Creek is pretty limiting there's a lot of limiting factors. It is a high mountain desert area, has a harsh environment. Temperatures in the creek are often exceed 21 or greater Celsius in the summertime. There's lots of forest fires, there's flash floods. It's a small tributary, and there's also a lot of natural barriers. So how did we start this study, or what are some of the methods? We tagged a bunch of we fished that were pit tagged, hatchery steelhead. We released them between the years of 2014 and 2019 in the spring runoff, primarily March and April, so peak runoff. And then we monitored their survival downstream from Omak Creek. And then we derived delayed migration estimates, 6 months, so 3 months to 6 months, 6 months to 12 months, 18 months and beyond, so up to 3 years after post-release. So there's a few different methods. Most people, when they're monitoring outmigration estimates, they use a CJS or a Cormack-Jolly-Sieber method. It basically looks at migration rates at a same time, same location, and a probability of survival to a known location. The GLS model, which is the Lower-Skalski model, it accounts for multiple sites and multiple years, recapture detections, and things of that nature. So we sort of followed that as well and used a modified CJS model. So we incorporated multiple years, and then we expanded those rates based on a tag rate and a detection efficiency from Omak Creek. So this is just a token slide on some model work that we did. I don't want to focus much on it. I will say survivals were pretty high. The variance was fairly low in our expanded estimates. But I really want to focus more on some of the factors and discuss some of the ideas and benefits of delayed migration. So here's some of the results. You can see that most of the fish that were delayed migratory fish went out in the spring of the following year. There was a few scattered that were actually 2 years and a couple that were actually 3 years beyond release. So then expand those numbers to the total release population, looking at tag rate and detection efficiency. And you can look at, there was anywhere from 17 to over 1,000 fish that were delayed migratory fish that went out after 3 weeks from release. So putting a percentage on that from total release, for each year, you can see anywhere from a little less than 1% to a little more than 3% of the population were actually delayed migratory fish, which may seem like a small number, but when you're looking at small tributaries, this has a huge effect. So the majority of the steelhead left post 12 months from release, similar to Buchanan and Skalski's findings. We determined anywhere between less than 1% to 3.5% migrated to the ocean 6 months or later. And this calculated anywhere from 17 to 1,352 additional fish to the outmigrating population. So just reiterating the table. What does that mean? So we release on average 20,000 steelhead annually into Omak Creek. And residual estimates can be anywhere from 5 to 8% based on mark recap studies in Omak Creek. So if you remove anywhere from 1 to 3% of those fish, you're potentially removing upwards of 1,600 juvenile steelhead from the residual population. This is pretty significant in a small tributary where you have all of these environmental factors, small water, less habitat, more competition. This is just I want to talk about some of the potential driving factors. This is a discharge in CFS, looking at what a fish might migrate at, and it varies widely, right? So we got anywhere from 16 CFS to over 170 CFS. So that may play a huge role. This slide, I'm going to say, just out there first, the correlation was okay between some of these factors, but there was really low confidence in a lot of it. So take some of it with a grain of salt. But just looking at from an eye test, on the left, there's delayed migratory fish, numbers of delayed migratory fish, which is colored in blue. The yellow line is also on the left, and it is, on the right, that's numbers of, total numbers of fish released in yellow. And then the black line represents the average discharge. So what you can see, or what it appears to me as that there is a positive correlation between the number of delayed migratory fish and I'm sorry, it's not mean flow, that's total or peak flow, just to clarify, is they move in the same direction, right? So when flow goes up, the number of delayed migratory fish goes up as well. So in essence, if you think of discharge as being habitat or increased habitat, there's more backwater, there's more side channels, et cetera, there's places for these fish to go if they're not willing to migrate in the first 3 weeks. Conversely, there's a negative correlation to the number of fish released to the number of fish that are delayed migratory fish. And you can see that in the yellow line compared to the blue line. So as there's more fish released, there's actually less migratory fish or less delayed migration. And if there's fewer fish released, there's more delayed migration, which was kind of a head scratcher to me, because seemingly you would think that the more fish you release, the more residuals, the more delayed migration, et cetera, et cetera. So I think this slide really shows that habitat has more of an impact than numbers of fish released. So some other food for thought, some genetic possibilities. Can delayed migration add to the genetic diversity or fitness of a return year? Think about it as, so you have fish that are going out from one parent group in 1 year, and all of their friends and relatives went out and they stuck around for another year, and then went out with a different set of parents that spawned the previous year. So what can this add to the genetic pool and the spawning population, similar to kelts that will repeat spawn and add to that population? Are we adding a different life history strategy that can be a benefit to the survival of steelhead? What are some of the take-homes here? Care should be taken when generating downstream survival using a fixed migration estimate. By not including delayed migration in your residual estimates, you may be increasing these numbers, which is a negative impact to hatchery programs. And our results also suggest that habitat availability is probably one of the primary drivers to migration timing and delayed migration. And then what are some of questions or something to pursue down the road? Well, what are some of the advantages to delayed migration? Is there a higher survival Because they're going out at a different time and not at a big group? Is there a greater adult return rate from these fish? Just something to look at. And can it increase the genetic diversity and fitness in a hatchery program similar to iterative parity? I want to thank everybody that helped. Grant PUD, the statistical analysis, Brooklyn Hudson, the field staff that tirelessly did all the work here. And then I've got a trivia here, name that band. If you grew up in the 80s, you might know, Should I Stay or Should I Go Now, right? So if anybody knows it, shout it out. But The Clash, right? I knew Tracy would know. All right, and with that, I'll say thank you.
All right, wonderful. At this point, I would like to invite our three speakers back up on stage, and anyone with questions, please either raise your hand to get the floating mic to you or come up to the standing mic here.
Yeah, I left my water here.
Thanks for the talks. Wesley, would your results maybe indicate that if habitat restoration was done downstream of a hatchery, you might unintentionally increase the late migratory counts?
Yeah, absolutely. Can you hear me? Okay, sorry. Yeah, we didn't measure or compare habitat restoration projects to the release. These fish were direct dropped above habitat projects that are done in Omak Creek. So that potentially could be a reason for them to hang up. We really just looked at the correlation to discharge and the amount of habitat based on water quantity.
Thank you.
I had a question for you. Yeah, so I had a question for you. Make it easy going back and forth with Mike. I was curious if you've looked at whether, since you found kind of the surprising result that the more fish you put in, the fewer residuals you're getting to some extent. Do you think that might mean that the residualism is being driven by the density of fish to some extent?
Well, that was certainly one thought.
Yeah.
And then when you look at the numbers of fish released, it wasn't density dependent. So in years where, and I don't have the slide up there anymore, but there was several years where, not several, but there was 2 years where there was the same number of, larger numbers of released fish, and the numbers of delayed migratory fish were very low. And then there was 1 year where there was a large number of released fish, and the number of delayed migratory fish was high. Conversely, there was also numbers of low release fish where the delayed migratory fish were low as well.
Okay.
So it seemed more driven by the higher flow, the discharge, than the available habitat.
Okay, thanks.
Kind of a combined question for Ben and Jason. Both of you sort of in one way or another estimated that hatchery fish seem to have lower fitness in some way than the wild fish. I'm wondering if, using two different methods, I'm wondering if there's a way to, I don't know, connect those two to provide some insight, for example, into, can you use the population model results to inform why the lower reproductive success is seen in the case of the parentage studies? And then secondly, I was wondering, in the case of the population models, can you, in effect, sort of use those to predict the viability of the population in the absence of hatchery supplementation?
Yeah, we could use the population models like that for prediction in general and for comparing different management scenarios, including the number of hatchery fish that you're putting in there. I think that one challenge with that is, at least when we're looking at the version that I was presenting where you've got all these environmental covariates, you don't know what those are going to be going forward into the future. But you can use them to help sort of tease out what you think the underlying dynamics of the population are and then run the model with just those, just kind of assuming an average environment going forward or tweaking the environment going forward if you want to. So you could certainly use it to look at that. And then as for the other question, there are a number of ways to kind of complexify a model like this, to expand it out, to start looking at some of the individual mechanisms or separating stages of survival, to where we could start to get at whether the differences in recruitment or mortality are linked to certain stages, or whether it's just genetic parentage or something else. Yeah, there's a lot of fun stuff to look at as we go, especially like we've got several data sets that are just kind of coming online over the last decade or more, and for an analysis like this at the population level, you get like one data point per year basically, and you need a long data set to really start to say something. And we've got a number of more complex pieces of data that are just kind of starting to grow into that size range where we can use them for things like this, so that's going to be very exciting.
Yeah, I don't have a whole lot to add, except that I haven't done any, I haven't added any kind of environmental covariates to our modeling efforts, other than having a year effect, I guess, is somewhat of it. But it would be interesting to try and add some of that in, for sure.
Hey, Ben, for your relative reproductive success for the reconditioned kelts, was that compared to all spawners or to kelts that were not reconditioned?
No, so that was compared to maiden spawning wild females.
Okay.
So first-time spawners that were wild.
Okay, so it was reconditioned kelts compared to first-time spawners.
Yep, so
Okay.
same comparison as for the hatchery fish.
Right, okay,
Right.
thanks.
Yep.
I beat you, Andrew. Anyway, just wanted to ask a question about the modeling in particular. We find that there's a lot of underlying data that has morphed in the last 10-year time frame. A lot of the fish data shows that same impact. A lot of climate data shows that same impact. And so I'm just curious, I noticed that the life cycle modeling stuff was done on a 30-year time frame, and there looked to be some correlation in kind of those early years, but it fell apart in the last 10. Are you doing any of your analysis based on just the last 10 years of information?
No, none of it's based on just the last 10, but one of the many things that I'll be doing, moving forward with this model is certainly going to be looking at the individual effects of these predictors that we get out of the full model on a year-by-year, effect-by-effect basis, and seeing, these things do seem to work really, really well early. Are they still holding up? So we can certainly break the time series of predictions down by individual effects, and that's something on my to-do list for this model.
Question for Ben. This, maybe I should know this, but your body lipid effect that you're seeing in your analysis, I think, is very intriguing, and I think you're one of the first ones to show that it has an effect on relative reproductive success. So my first question is that body lipid variable, the way that you're measuring it, is it correlated to any other fish metrics that you've examined? And if not, then I want you to speculate on how body lipid content is influencing relative reproductive success, both in females and in males.
All right. Yeah. So
And you'll be graded.
I have looked at, you know, if there was a relationship to like a condition factor or to length or things like that, and it wasn't really popping out that there was obvious relationship there. But I probably haven't exhausted, you know, the search. And as far as like why we expect that lipids would have an effect, it's stored energy, right? And we're measuring it in the muscle tissue. So there's a Penny and Moffat wrote a paper kind of describing how lipids are exhausted in the muscle tissue during migrations, and that, you know, having more lipids when they finally get to the spawning grounds is probably critical for being able to maybe dig a bigger, deeper red or wait for a mate for females, and then for males maybe just having more energy to compete with other males. And I also wanted to say they did also down in the Kalama, they did see the same difference we saw between hatchery and wild fish for the fat content. So it's not.
So I thought, follow-up question, I should have sat down. So if it is really important, and it is, and we're talking about parents, then the effect would be happening, the difference is occurring very early in the life stage? Or do you think somehow that effect is translating into juvenile or adult life stages? Or do you think it's really specific to how well you build and guard and compete for space and females?
Yeah, that'd be my guess, the latter, that it's just at spawning that where that fat content's important. As to why they have a lower fat content, I'd have to speculate a lot there. I don't know.
And I can't remember, for your hatchery fish, do we have hatchery fish broken out by wild by wild, wild by hatchery, hatchery by wild, to see if there's a broodstock effect associated with body lipid content?
Yeah, so both the Wells and Twisp stockfish had similar lipid levels, which was below the wilds.
But did you see any broodstock effect, like if you had a wild female and a hatchery male versus two hatchery parents? You know what I'm saying?
Right. So we, yeah, I don't know offhand of an effect like that.
Okay.
But could probably dig into it more.
Just the other work that's been published, we see that kind of a different broodstock effect in that the origins of the parents matter, not necessarily what kind of broodstock it was, but you know, hatchery by hatchery fish, progeny of hatchery by hatchery had the lowest relative reproductive success. Wild by wild has the highest, still lower than wild fish. But I'm wondering if the lipid content, is there some genetic component associated with hatchery parentage that you would see in your data? Or do you have enough information? Do you have enough like hatchery by wild versus hatchery by hatchery fish to tease that stuff out? So we would go across your kind of different broodstock the different broodstocks that we've used, unfortunately.
Right, and that's the main source of that.
Yeah.
That we have. Like we have a little, you know, a small percentage of our adults have both known parents for wild spawners, if that's what you're getting.
I would just be talking about the hatchery fish.
Yeah, and so we do have that for
Okay.
the you know, the well stock is all hatchery by wild.
Oh yeah,
Crosses, right, and the
Yep.
Twisp is wild by wild.
And it's all wild by wild. And
I
do we
mean,
have
pretty
anything in the
much.
middle? We don't have any middle.
Well, we don't have the bottom end. We don't have the hatchery by hatcheries to compare.
Isn't that the well stock?
They're hatchery by wilds.
Oh, were they?
At least they were at the time when
Okay.
we were
Gotcha.
including them
Okay. All right. Thank
for
you.
the most part.
Two things for Ben. First of all, I really liked your presentation. It was really good information. I was curious, have you published these results any place where we can get our hands on this information in a more...
So we do have a annual BPA report, but we're working on a manuscript now.
Okay.
For this
Love to see that.
information.
Number one. Number two is, well, I've forgotten it already. Oh, well. Wasn't that important.
We have time for one more question if anybody wants to make their way to the microphone. Oh, we have a taker.
For those reconditioned kelts, was that just, were they hatchery fish, or were they wild, or?
I believe they were all wild. There may have been one unmarked fish that came out as hatchery, but mostly they were wild females.
Okay.
For the Twisp program.
Speculating, do you think if you took a hatchery fish and put it up in a luxury hotel for a year and fed it krill, and do you think you would see the same higher, kind of spawn rate that you saw out of the wild?
Yeah, it kind of depends on what it looks like that lipid content is really affecting their their reproductive success. But maybe for a hatchery fish that gets, reconditioned, they might have less of an effect, but I suspect it would still be some.
So there is something strongly genetic there. Because if you put them both in the same scenario for a year, you would still expect them to put on different lipid content.
Potentially.
Yeah.
I don't know that it's necessarily genetic, though. It would just be, that their life prior to that might matter as well for what it's done to their physiology or whatever,
Sure.
All right, thank you very much. If we can all thank our speakers again for their great answers. And with that, we are done with our first session, so thank you for sticking around. We will have a lunch break from 12 to 1 p.m. with our next speaker starting at 1 p.m., so please try to get back a few minutes before that so you don't miss that talk. Thanks, everyone. Thank you, guys.